Make it to the lectures we went to on red days, Monday and Tuesday after Thanksgiving. And I'm going to introduce the second section of Chapter 7, which deals with the plasma membrane. Plasma membrane is very important to us because it helps the cell to maintain homeostasis. And it does that because it defines the outside edge of the cell and controls what goes into and out of the cell. So we spent a lot of section 1 learning about how the cell was discovered with the invention of the microscope, and that was a very important discovery, not only for science, but it really changed the way people looked at the world. But it was a relatively new discovery. The mid-1800s or close to 1900 before we had really good microscopes and understood the cell, and we've come a long way since then. We now know a lot more about the inner workings of that cell, including structures such as the plasma membrane. Your objectives for this section are to be able to describe how a plasma membrane functions, and I like to point out which of the objectives I would like to think are the most important to you. Well, in this case, the most important objective you have is to identify the role of proteins, carbohydrates, and cholesterol. And I also include the phospholipid bilayer itself in the plasma membrane. If you understand what the jobs of each of those are, you will have a good idea on how the plasma membrane functions. So the plasma membrane is a border, a barrier, a wall around the cell. And if we wanted to build a wall around a cell, there's some things we need to keep in mind. Certain things are going to need to get into that cell, things like water and food. What else do we need to get in? What else would we need to get in? We might include oxygen in that list. What do we need to get out? Wastewater, solid waste. Things we need to keep in would include DNA and proteins. So we want a membrane that isn't a perfect barrier for everything. Some things need to get in, some things need to get out. But not everything needs to get in and get out. And it needs to be more than just semi-permeable. It needs to be selectively permeable. Because the real function of the plasma membrane is to maintain homeostasis, to keep internal conditions relatively stable by adjusting them by letting things in and letting things out. And we can't do that if we don't have a defined space. So the plasma membrane helps us define the space we're going to maintain homeostasis in, and then it's going to actively participate in keeping the materials where they need to be. So we're going to need to get in things like food, water, and fuel, as well as information, and we're going to want to let out information and waste. What we are really doing when we do these things, what we are saying without coming out and saying it, is we are maintaining homeostasis. The definition of homeostasis is to keep internal conditions just where they're supposed to be. And we do that by getting rid of excesses and allowing in materials that we need. So we do this using selective permeability. Selective means the ability to choose. So we choose what goes in and out of the membrane. And permeability means we have the ability to pass through it. So we have the ability to choose what goes in and out of a selectively permeable membrane. We can make that selectivity based on size and shape. So there are some things that are just too big to fit between the phospholipids of a plasma membrane. There are other things that are just right. They'll get in for different reasons, and by passing through the lipid bilayer. And there are other things that might be similar in size, but their shape is wrong, or they have a charge, and those things can't get past the phospholipid bilayer. So this size and shape allows for some selectivity. More importantly, we have specific selection in many cases. So if we want a specific thing to come in, like a sugar for instance, we will have a receptor that will allow that sugar to specifically come through the cell membrane. So our selective permeability can be based on size and shape, or it can be based on specific selection. We said from the beginning that structure is always linked to function. I just went over the functions, selective permeability, and told you how that selective permeability comes about. So we're going to now take a look at the 4 structures that allow that plasma membrane to be selectively permeable. The first component is going to be the phospholipid bilayer. The phospholipid is made up by layers made up of phospholipids, phosphate heads on the top, lipids underneath, and then the second layer, the phospho heads, are on the bottom and the lipids are in the middle, giving you the phospholipid bilayer, polar head and 2 tails, 2 layers of phospholipids. Their function is the barrier function. The function of the phospholipid bilayer is to keep water, to keep 2 water environments separated. When you set up a structure like this, water is forced upward and water is forced downward. So this would be the water on the inside of the cell, what we would call cytoplasm, and this would be the water outside the cell, which we would call interstitial fluid. So that phospholipid bilayer provides the bulk of the barrier to 2 water environments. The second structure is going to be the transport proteins. Transport proteins are going to allow specific materials into and out of the cell. So we have a phospholipid bilayer that's providing a barrier separating 2 water environments, and in that phospholipid bilayer we're going to place a transport protein, and that transport protein is going to specifically move materials from one side of the membrane to the other. And they are specific materials. So transport proteins are going to allow for the entry and exit, depending on how they're oriented, of specific materials. And now we've got some selective permeability. The third component of the structure is cholesterol. Cholesterol is going to provide for fluidity and strength. It's going to help hold the membrane together, but it's going to help allow things in the membrane to move about more freely. So here's our phospholipid bilayer separating 2 water environments, our transport protein that is making this semi-permeable membrane selectively permeable, and within that we're going to put cholesterol molecules, and those cholesterol molecules are going to give us strength and fluidity. So in reality, that transport protein is not going to be stuck there. It's going to float all around this way and that way, and because it's 3-dimension, into and out of the screen. The fourth component is carbohydrates. Carbohydrates provide information. They supply information to other cells, and a lot of that information is identification information. So they identify your cells as being your cells. So each of us has on our cells signs that say those cells belong to us. In our immune cells, the cells that protect us from diseases like COVID or bacterial infections, look around our body for cells that don't have our signs on them. And when they don't have our signs on them, like a cell infected with COVID or a bacterial cell, these immune cells will kill that cell without our sign, which is very good for getting rid of diseases. But it can also cause a problem to us if we need to transplant an organ between 2 individuals. Because if you needed a kidney from someone, let's say Joe was going to give you a kidney, the kidney you got would have Joe's signs on it, not your signs, and your immune system would think it's being attacked by a huge bacteria. So it would try to kill that kidney, even though you needed it to stay alive. But we have our membrane here with a phospholipid bilayer separating 2 water environments, made an even better barrier because cholesterol doesn't like water, and made more fluid so that things can move around in the membrane, and selectively permeable because of these transport proteins. And now we're going to attach some carbohydrates to some of the structures in the membrane to provide information to other cells, especially in multicellular organisms. So those carbohydrates provide identification information. If we take it all together, we end up with what scientists call the fluid mosaic model. It's called the fluid mosaic model because those four things we just talked about behave as though they were in a fluid. They're not cemented in place. They're moving around freely from left to right. Why is it a mosaic? A mosaic is something made up of different parts, and we have four different parts that make up a mosaic: carbohydrates, transport proteins, cholesterols, and phospholipids. And we call it a model because it describes what we know about the cell membrane up to this point. So if this was truly a fluid, you would expect that things in it would move. So if this green dot was a fluid, you would think that if the wind blew, it might move. If a wave came up, it would move somewhere else and continue to move as long as there was something happening to it. And that would be what would happen to something in a fluid. And when we look at membranes of cells under microscopes, we can see the parts of that membrane moving around freely. So it behaves, the things in it behave like they were in a fluid. Again, it's a mosaic because it's made up of different parts. The four different parts here are four different tiles are red, blue, green, and gray to make up the image of a fish. And we have tiles that make up the image, or we have parts that make up the plasma membrane. We have phospholipids making up the bulk of the barrier. We have transport proteins making it selectively permeable. We have cholesterol molecules making it more fluid and stronger. And we have carbohydrates giving identification information to other cells. And it's a model. Just like this model of a DC-10 gives us information. We can make predictions about how many people could fit on a DC-10, what kind of fuel it would use. We could compare it to other models to get an idea of size. It doesn't tell us all the information, but it explains what we are looking at. So we call it a fluid mosaic model, which is a representation of the cell membrane composed of different pieces, and those pieces can move freely within the membrane. The fluidity comes from the fact that those phospholipids behave like they are a liquid, not like a layer of cement. And the mosaic is because it's made up of different molecules in the membrane. Here's an artist's drawing of the fluid mosaic model. We have the outside of the cell, the inside of the cell. This would be cytoplasm. And the bulk of this membrane is made up of phospholipids, the phospholipids in orange, the phosphoheads on the top and the bottom, and the lipid tails together in the middle, providing a bulk of water barrier. There are transport proteins shown here as globular proteins that allow for pathways through the membrane for specific materials, making it selectively permeable. There are cholesterol molecules embedded within the lipid part of the phospholipid bilayer that make it a better water barrier, but also make it more fluid, so that this protein might float all the way over to here, and this one could come over here. And then lastly, there are carbohydrates attached to that, and those carbohydrates provide identification information. So in summary, the selective permeability is a property of the plasma membrane that allows it to control what enters and leaves the cell. The plasma membrane itself is made up of two layers of phospholipid molecules. That's why we call it a phospholipid bilayer. There also contains cholesterol and transport proteins. Cholesterol makes it a stronger, more fluid barrier, while transport proteins make it selectively permeable. And collectively, we use all this information to describe the plasma membrane in the fluid mosaic model.